Good day, everyone. We're reviewing Wetzel's Limnology textbook. This is the fourth edition, and we are on chapter 11th, which deals with oxygen and how oxygen cycles through limnological system, lotic and lentic systems. And today we'll be specifically looking at photosynthesis and photorespiration. But before doing so, let's look at figure 11-3. Effect of organisms on oxygen transports in sediments. So we have three figures here. We have A, B, and C, and three different processes. So A is oxygen penetration depth in several marine and freshwater sediments, where transport is either almost entirely controlled by diffusion, white bars, or greatly enhanced by advective transport to the burrows of fauna, shaded bars. And these are the species of fauna that we're talking about that are mediating the transport of oxygen in the subsurface. And this is, if you look at this distribution here, so a northern Pacific, for example, that's an ocean, of course, and it goes from the northern Pacific to Lake Vecten, so maybe that is in Switzerland or in Germany somewhere, or in Austria, or somewhere in Central Europe. And so what we see here is we see so the y-axis is the depth, and then the x-axis is the location, and so the y-axis is logarithmic in scale, and it doesn't really give us like a concentration of oxygen per se, but just how deep oxygen, I guess, probably is detected. So in the Pacific or Northern Pacific, for example, it's detected at about 200 or 300 millimeters, depth, where in Lake Vecten, it's only detected at 0.1 millimeter depth. Okay, so there's a quite variable value here. It's like 3 times the range in the oxygen penetration at 3 times. And then here we can see that organisms also have an influence upon oxygen distribution in the subsurface. For example, like Sialanassa is allowing oxygen to go down to a depth of about 1100 millimeters, and this is Caronomus, and that is lower. That is about maybe like 150 or 200 millimeters. And then here they're saying the bar for Caronomus correspond to a freshwater example. So this is freshwater, and this is probably saltwater. So B looks at bioturbation, so which is the process of like sediment-based animals such as worms or invertebrates like that to mix the sediment through their life cycle. As they are moving in the subsurface, they are allowing some amount of respiration to occur and some amount of ventilation of water that is oxygen rich to reach areas, subsurface areas in the sediment. So it's not the Caronomid larva that is producing oxygen, it's more the ventilation that's producing oxygen. So the other thing is the presence of that larva is also enhancing bacterial respiration. Unfortunately, though, it's enzymatic thing is generating carbon dioxide. And this in light yellow is the oxidized sediment, and then in the dark yellow is the reduced sediment. So it's 2 different states. And so as we can see, ventilation is definitely generating a fair amount of oxygen, or allowing a fair amount of oxygen to off gas, I guess, into the sediment from water. And then this is, I guess, a typical like wetland plant. So we have flood water here. We have the root system of that plant in the sediment, and the root system, below the flood waters. And then what is interesting is that that plant is producing oxygen, and that oxygen gets transported in the subsurface and is off-gassing through their roots. And then, but it's uptaking methane in the soil, and the reverse is true. Methane is then off-gassed by the leaf, by the parenchyma, I think that's what I'm saying, which is a structure in a plant. It's off-gassing methane in that case. Okay, so that was an introduction to that photosynthesis and photorespiration. Oxygenic photosynthesis is the fundamental source of dissolved oxygen supersaturation in productive inland waters. The availability of light and nutrients for photosynthesis is the net result of interacting biogeochemical processes acting at differing spatial and temporal scales. In response to these drivers, the photosynthetic production of oxygen in inland waters varies across multiple orders of magnitude from 10 to the minus 3 to 10 to the 5 nanomoles of oxygen per meter cube per hour, showing higher values for benthic primary producers and macrophyte. As a reference, the highest rate of production, 10 to the 5th millimoles of oxygen per meter cube per hour, so this is not nanomoles, millimole, sorry, would bring a volume of anoxic water to saturation at 0.37 millimoles of oxygen per liter in 10 seconds. Expressed as an aerial rate, photosynthesis in phytoplankton and macrophyte reaches maximum values of 80 millimoles of oxygen per meter squared per hour. Photorespiration is the light-dependent oxygen consumption by the enzyme ribisco in photosynthetic organisms. Photorespiration rates in freshwaters have been considered low in comparison to rates in terrestrial plants due to the low relative oxygen solubility in water and the presence of carbon concentration mechanism. However, high photorespiration rates, accounting for 25% of net photosynthesis, has been observed in macrophytes, suggesting that this mechanism could be important in some situations. Aerobic respiration. Aerobic respiration of organic matter is frequently the most important oxygen-consuming process in freshwaters. Aerobic respiration and photosynthesis together are the dominant biological mechanism shaping oxygen concentration in freshwaters. The use of oxygen as the electron acceptor for oxidizing organic matter always yields the highest amount of energy compared to any other receptor, and the presence of oxygen suppresses most anaerobic metabolic pathways. The dramatic changes in aerobic and anaerobic respiration metabolism across oxyclines in water and sediment thus become fundamental for matter and energy cycling in aquatic ecosystems. Similar to photosynthesis, respiration rates in freshwaters respond to a complex combination of biogeochemical processes acting at differing spatial and temporal scales. In lakes, respiration usually does not show the large variability shown by photosynthesis, but it may be more variable than photosynthesis in rivers. However, different methods reveal different respiration rates. For example, bottle incubation yields volumetric respiration rates in the water column for 10 to 4,000 millimoles of oxygen per meter cube per hour, which correspond to aerial rates between 240 and 4,500 millimoles of oxygen per meter square per hour. Sediment core incubation showed a range from 40 to 800 millimoles of oxygen per meter square, and whole ecosystem respiration rates estimates derived from open water diel oxidation curve reach about 24,000 millimoles of oxygen per meter square per hour. Methane oxidation is a type of aerobic methylotrophic metabolism. Methane diffusing from anoxic sediments from rising methane bubbles or produced in the water column is oxidized in the presence of oxygen by methane oxidizing bacteria. Methane oxidation usually occurs in oxyclines in the freshwater column or sediments as well as the hyporeic zone of rivers. Freshwater methane oxidation rates range from 0 to 833 mole of methane per meter cube per hour and can result in the consumption of up to 98% of stored methane in a water body during seasonal mixing. Oxidation of reduced inorganic compounds. The oxidation of reduced substances can be an important contribution to the oxygen budget of water bodies if they are contrasting redox conditions across a spatial gradient. The sediment pore water and hyporeic zone can be enriched with reduced ions that consume large quantities of oxygen, especially when released into the oxic water column. A rich mosaic of biogeochemical processes is involved in the oxidation of reduced compounds with oxygen. Almost invariably, sediment-water interaction largely control the amount of reduced substances available for oxidation in the water column of lakes and rivers. Oxidation of iron and manganese, ferrous iron, Fe2+, oxidation involves both biotic and abiotic reactions. But as a simplification, biotic processing of ferrous iron outcompetes abiotic oxidation of low oxygen and high ferrous concentrations. These conditions are typical at chemoclines with opposing gradients of oxygen and ferrous iron. For example, in river biofilms and in the water column of meromictic lakes. In contrast, manganese ions oxidation is mainly mediated by biological activity, as the abiotic oxidation of reduced manganese, Me2+, is very slow at pH values usually found in inland waters. Oxidation sulfur compounds, reduced sulfur compounds, mainly sulfide and elemental sulfur, are oxidized mostly by biologically mediated reactions. Bacteria and archaea comprise most of the microorganisms that oxidize reduced forms of sulfur in large quantities, 50,000 macromoles per liter per day. The oxidation of sulfide can revert the sediment from a sink to a source of sulfate to the overlying water seasonally, particularly in oligotrophic systems where oxygen penetrates deep in the sediments. In highly productive sulfate-rich lakes, sulfide can be released into the anoxic water column and be oxidized upon mixing, accounting for a substantial share of oxygen consumption in the hypolimnion, even causing fish kills. So why don't we look at figure 11-4 here. Monthly dissolved oxygen concentration at the surface waters of Sal Reservoir in Spain during 7 years eutrophication. So y-axis is the surface oxygen concentration in milligrams of oxygen per liter, and then the x-axis is the time in month. So, and we're looking at 7 years from January 1984 through January 1991. And what we see is we see the sort of yearly variability in the concentration of oxygen at the surface, at the water surface. And so what we have like higher oxygen concentration like in the mid part of the year, and then lower oxygen concentration in the winter season or the late fall, early winters. So note the extremely high values during summer due to primary production, and the extremely low values during fall turnover when the oxidation of reduced compounds from the hypolimnion, mainly ammonium, consumed almost all oxygen. This is an example of how intense oxidation of reduced substances can create anoxic conditions even in well-mixed surface waters. A common threshold for defining anoxia in freshwater is less than 1 milligrams of oxygen per liter. It is indicated as a horizontal dashed line for reference. So this is the horizontal dashed line, and so below that, it's we're dealing with anoxia pretty much. So we have the absence of oxygen in pretty close to it. All right, so oxidation of ammonium. Although abiotic ammonium oxidation has been measured in soils, nitrification is considered a biologically mediated process in inland waters. If large amounts of ammonium are present, nitrification can consume large amounts of oxygen. Nitrification is partially responsible for oxygen consumption in the hypolimnia of lakes during summer stratification and under ice, and it is responsible for anoxic conditions in rivers downstream from point sources rich in ammonia. Oxidation of hydrogen. Aerobic hydrogen oxidizing bacteria usually thrive in the epilimnia of lakes where hydrogen that has escaped from the sediments and hydrogen produced by nitrogen fixation in the surface layer can accumulate However, the concentration of hydrogen in the oxalic layers of fresh waters is usually orders of magnitude lower than that of methane, and therefore hydrogen oxidation rates likely do not consume relevant quantities of oxygen. Photochemical oxidation of organic matter. Reactive oxygen species, singlet oxygen superoxide and hydrogen peroxide, formed by the photoexcitation of organic molecules, can also react with organic matter consuming oxygen. Although photochemical oxidation processes are likely masked in very productive waters by intense respiration, photochemical reactions drive oxygen dynamic and affect metabolism estimates in fresh waters with high concentration of chromophoric organic matter. For instance, photochemical oxidation in highly colored lakes can be as high as 0.12 milligrams of oxygen per liter per hour and can result in substantial carbon dioxide production in the surface layers of many freshwaters. Chemical weathering. Rocks containing chemically reduced minerals, for example, olivines, pyroxenes, amphiboles, metal sulfides, undergo oxidations when in contact with oxygenated waters. However, the rates of mineral oxidation from bedrock or regolith are deemed too low to have a discernible effect on oxygen concentration in most surface waters. Chemical weathering likely only has a measurable impact in water bodies with very long water residence times and where other oxygen consumption mechanisms are very low, for example, amictic lakes. Distribution of dissolved oxygen in running waters. Flow is the major structuring force in rivers. In this chapter, we refer to rivers as running waters of any size, from small headwater streams to large lowland rivers. The flow of water in rivers maintains a constant supply of nutrients, produces physical turbulence that constantly mixes the water column, promotes reoxygenation via atmospheric exchange, and results in frequent scouring that limits permanent sedimentation. Water flows along the surface of river channels and through sediment and rock interstices within channel boundaries. So, for example, aporeic, parafluvial, and riparian zone to the stream and vice versa. These lateral and vertical pathways are also involved in both the advection of dissolved oxygen as well as other substances, indirectly influences riverine dissolved oxygen dynamics. In addition to this spatial variability, oxygen concentration in rivers may vary temporally on diel, seasonal, annual, and decadal scales. This rich spectrum of temporal signals on multiple scales combines to shape the oxygen dynamics of a river, which integrate the biogeochemical processes occurring within the river and its catchment. Spatial variation. River network scale. River hydrodynamics, geomorphic characteristics, and watershed land uses generally change in predictable way from small headwaters to large rivers, as these attributes largely determines the physical dynamics and biogeochemical processes driving the content of oxygen in water. The river continuum process can be used as a simple hierarchical framework to provide general expectation for dissolved oxygen concentration along river networks. Slope and flow velocity correlates with factors such as river depth, surface area, water residence time, and temperature to influence atmospheric exchange. Because high reaeration can override signals of riverine metabolic activity and thus limit low oxygen conditions, the oxygen content of small headwater stream is expected to be stable and approximately at or somewhat above saturation. However, biotic consumption of oxygen and reduced solubility as a response to increasing water temperature is more pronounced in smaller streams than in large rivers, leading to a more variable oxygen concentration in low gradient headwater streams that are less influenced by gas exchange. Conditions of increased temperature in smaller systems are usually associated with anthropogenic water extraction, river impoundment, or riparian tree cover loss, but can also occur during flow stagnation or extended low flow periods during droughts. Droughts and flow intermittency are common to many rivers worldwide, a phenomenon that may become even more widespread with global climate change. As flow recedes and river networks contract, water residence times and temperature increase, and as a consequence, oxygen concentration decreases. So before we move on, let's look at this figure here. Figure 11-5. So conceptual model of patterns and drivers of dissolved oxygen concentration along the river continuum from headwaters to large rivers in an idealized river network. The interaction between factors associated with channel hydrogeomorphological dynamics, watershed land uses, level 2, and riverine physical and biogeochemical processes, level 3, generates expectation for patterns and variability of dissolved oxygen saturation along river networks. So level 1, let's see here. So hydrodynamic and geomorphology. So this is the process we're looking at. Slope and flow velocity. So when slope and velocity predominates, depth and width of water residence time and temperature are not as much of a factor in terms of oxygen distribution, I believe. Watershed land use, natural versus urban agricultural. Processes affecting river dissolved oxygen, river atmosphere exchange, solubility, advection, metabolic activity and diversity in pathways, and then dissolved oxygen, atmospheric equilibria. And then we have the headwaters, the medium-sized streams, and then large rivers here. So this is like your typical river system. And as they are saying, this is a river continuum, this is a concept. Okay, I think that's going to be it for today, and we'll pick it up tomorrow with this section right here, in page 246, in contrast, large rivers. Thanks for sticking with me, and we'll talk tomorrow, and I'll see you tomorrow. Thank you. Bye.